Good morning. I'm Dr. G. Vijay Pratap, lecturer in zoology, Dr. V. Krishna Government Degree College. I welcome you all to this prestigious online platform called LMS, which is specially designed by our special commissioner, Srimam Naik Garu, as well as the team of the Commissionerate of College Education. Now let us quickly learn about the title, what you are going to learn today. The title is Health Management in Aquaculture, Diagnostic Techniques and Quarantine. The first learning outcome is student will be able to learn about the different diagnostic techniques in the field of aquaculture, and the student will be able to learn about different quarantine procedures in aquatic animals. Yes, let us go through the introduction. We all know that aquaculture ranks second position in the world fish production, and it contributes a lot to Indian economy. And it offers employment to lakhs of people either directly or indirectly. But disease outbreaks in these sectors usually fetch huge losses if they do not tackle timely. There are a number of diagnostic techniques used for health management in aquaculture. Some of them are case history, clinical signs, necropsy or postmortem, microscopic diagnosis, microbial diagnosis, biochemical diagnosis, and immunological diagnosis. Finally, molecular diagnosis. Let us quickly go through the case history. Case history information is very necessary for proper diagnosis and for proper administration of medicines. Some of the case history parameters that can be taken into account for the purpose of preparation of case history are study of fish and study of environment. Under study of fish, we could study the age of the fish and size of the fish, mortality time and mortality rate. Under the environment, we could study the physicochemical parameters like the salinity, temperature, DO, nitrates, phosphates, whatnot, everything. And microbial load also studied, and especially the facilities which are provided in the rearing unit are also examined. Let us go through the clinical signs of diagnostics. Clinical signs such as weight loss, changes in feeding behavior, lethargy, parasitism, erratic movement, discoloration, escape reflection, flashing or itching, hemorrhagic lesions, ulcers are considered to be the first sign of disease related problem or a stress related problem in the aquatic system. Coming to the necropsy or postmortem. The necropsy examination is performed to enable us to know the root cause of why the disease has erupted. Let us see the microscopic diagnosis. We know pretty well that microscopic diagnosis is always considered to be the best reliable tool in the aquaculture health management system. Microscopy could be a quick tool, but the accuracy depends on the experience of the microscopist and the quality of the equipment. That's the most important point. There are two types of microscopes are used in the aquatic system. The first one is called light microscope. The second one is called electron microscope. The electron microscope again divided into two types: the transmission electron microscope, scanning electron microscope. Light microscopes can be used for observing even the small scratchings also, but the electron microscopes are used for observing changes in the tissues at higher magnifications. Now let us quickly enter into the another diagnostic tool called microbial diagnosis. Let us quickly go through the microbial diagnosis. There are two tools available for the microbial diagnosis. The first one is called staining method. The second one is called motility test. Again, staining method divided into two types. The first one is called gram stain method. The second one is acid fast staining method. Coming to the motility test, it also can be divided into two types. The first one is hanging drop method. The other one is agar stabbing method. Let us quickly know about all these methods in detail. Coming to the gram stain method, it was created by Hans Christian Gram in 1884. Apparatus and chemicals that are needed for conducting this experiment: a clean grease-free slide, a primary stain called crystal violet, secondary stain called basic puxin, grams iodine, decolorizer 90% alcohol, and nichrome wire loop, and finally bacterial suspension. Let us go through the entire procedure. Take a clean slide, wash it and dry it, and with the help of loop, prepare a smear on the slide and dry it and heat it to fix the slide, and then add few drops of crystal violet, allow it for 2 minutes. After that, wash it and dry it, then add iodine of few drops and allow it for 2 minutes, wash it and dry it, and then add decolorizer and wash it and dry it. After that, the secondary stain like saponin is added, then wash it and dry it. After that, you can observe the slide under the microscope. You see that the cells appeared with crystal violet color are called gram positive cells. The cells appeared with pink color are called gram negative cells. Coming to the acid fast staining method, you see that apparatus and chemicals, clean grease-free slide, primary stain like Gill Nielsen carbon fuction, it is in pink color, and bacterial cell suspension, a decolorizer like acid alcohol, and 1% malachite grain. Now, let us quickly learn the procedure of acid fast staining method. Just take a clean grease-free slide, and with the help of loop, you prepare a smear on the slide, and then add ZNFC stain. After that, wash it, then add acid alcohol. After that, wash it, then add 1% malachite green, then wash it and dry it. Now you can observe it by keeping it under microscope. Acid fast bacteria appear in pink color, non-acid fast bacteria appear in green or blue color. This is the procedure. Let us quickly learn about the motility test. Motility test used to find out the microorganism, a motile one or a non-motile one. And there are two methods, I already told you, the hanging drop method and agar stabbing method. Coming to the hanging drop method, apparatus and chemicals which are needed: depression slide, cover slip, bacterial cell culture, inoculation loop, petroleum jelly, and Bunsen's burner and laminar flow. Let us see the procedure of this method. Take a concave slide. You see, observe the concavity in the middle of the slide. And now we take a cover slip Just with the help of pipette, you place a drop of bacterial culture on the cover slip. Exactly, it should be exactly like a pearl. And then you place this then you place this concave slide. Just this is the concave slide, and you place this concave slide exactly on the cover slip, so that the bacterial culture which is present here is exactly located in the concavity of the slide. Then the petroleum jelly which is applied around the brims of this cover slip slide, so that there will not be any leakage. Now you invert the slide. You see that now you inverted the slide. You see that this is the position after the inversion of the slide. This slide can be used under the microscope to find out whether the microorganism is motile or not. Let us see the second motility test, agar stabbing method. Apparatus and chemicals. Sulfide indole motility medium, which is called SIM, bacterial culture, inoculation needle, Bunsen burner, laminar flow, and cotton. Let us quickly go through the procedure of agar stabbing method. Just you take a bacterial culture. After that, with the help of inoculation needle, you take the bacterial culture, a few drops of bacterial culture, and place it in the SIM solution with the help of a loop or inoculation needle. Then you keep the SIM solution into the incubator at least for 24 hours at 37 degrees centigrade. After 24 hours, you find that a black precipitation in any test tube which shows the motile positive bacteria, and you see this is black precipitation which shows the motile positive bacteria, motile, non-motile. Now let us go through the biochemical diagnosis indole test. Apparatus and chemicals. 1% of tryptone breath, Covax reagent, incubator, test tubes, and inoculation loop. Procedure. You take 1% tryptone breath and you place the bacterial culture with the help of inoculation needle into the tryptone breath and incubate it in the incubator for 28 hours. And after that, you take it out and add Covax reagent into that solution. Then you see the observe it. If red precipitation is formed, it is called indole positive. If the light yellow precipitation is formed, it is called indole negative. Let us see the catalase test. Apparatus and chemicals. Clean grease-free slide, hydrogen peroxide, glass capillary tube, Bunsen burner, marking pen, inoculation loop, water and laminar flow, bacterial culture. Let us see the procedure. Just take a clean grease-free slide and mark it with the help of marker. The marker can be called, one side can be called as T, other side can be called as C. Then place a few drops of water in the circles Then take the bacterial culture, and with the help of inoculation loop, you take some bacterial culture and place it on the water drops of the slide. Then after that you add hydrogen peroxide. Then you observe it. If the bubbles are formed, that is called catalase positive. If no bubbles are there, that is called catalase negative. Let us quickly go through the immunological diagnosis. Serological tests can be performed for diagnostic purpose when a pathogen is suspected. In agar gel precipitation test, we are going through the agar gel precipitation test, and it is used to determine the immunoglobulin levels, sufficient antibodies and antigens placed in the wells of agar plates. So antibodies placed in the center well and antigens placed in the surrounding wells. When particular antibodies and antigens meet each other, a white precipitation line is formed in between the walls of two wells. By observing these precipitation levels, we can assess the immunoglobulin levels of the organism. Though this is the best method for immunoglobulin estimation. Coming to the molecular diagnosis. So in this test, polymerase chain reaction is there, and in this technique is used to amplify the DNA strand. Entire process done in a machine called PCR machine. In the PCR machine, there are 3 steps involved. They are called polymerase chain reaction. The first step is called denaturation. This process breaks the double-stranded DNA into 2 strands of DNA. And the second process is called annealing. It helps to form the new DNA strand by copying it. 3 extension, repetition of denaturation and annealing process in order to get more copies of DNA strands. Let us learn the quarantine. Quarantine is the method to isolate the sick or suspected sick animal for observation and rejuvenation. That is the first. And the minimum quarantine period depends on the several sets of quarantine protocols framed for an imported aquatic animal or for a local aquatic animal. Quarantine can be observed at the international level, subnational level, and at the production facility level. The basic requirements for effective quarantines are: 1. Adequate physical infrastructure, established operating protocols, well-trained staff, and biosecurity management. Now it is the time for what we have learned today. We have learned about case history, clinical science, and necropsy, and we have learned about microscopic diagnosis, we have learned about microbial diagnosis, and we learned about immunological diagnosis, and we have learned about molecular diagnosis, and we have all and you have also learned about the quarantine methods and procedures. If you want to learn more in detail, you go through the CC website where the e-content or text in detail is especially provided for you. Please go through it, learn it, explore it, and stay blessed. God bless you.